Hi, I'm Dr. Mike Darcy, and today I'm going to be talking about bacteria, archaea, and viruses, together with a brief explanation of viroids and prions. So let's get started. Okay, right. So, bacteria, archaea, and viruses. So, what are we going to talk about? We're going to start by talking a little bit about prokaryotic cells. There's two types, major categories of cells. There are prokaryotic cells and there are eukaryotic cells. Prokaryotic means before nucleus, and eukaryotic means true nucleus, both from Greek. Um, so, prokaryotic cells do not have a nucleus. We are eukaryotic cells. Well, we are not eukaryotic cells. We are composed of eukaryotic cells. All animals are, all plants are, all fungi are, which means we basically have a nucleus in the center of the cell, and that's where the DNA is kept in the form of chromosomes. Prokaryotes do not have a nucleus. The DNA is just floating loosely in the cytoplasm, the liquid of the cell. Major difference between prokaryotic and eukaryotic cells, other than the fact that prokaryotic cells do not have a nucleus, is the fact that prokaryotes are so much smaller, thousands of times smaller than eukaryotic cells. They're tiny, which means they can replicate quite quickly. So they can grow and divide and become quite invasive in a relatively short period of time. Some prokaryotic cells, for example, can divide once every 20 minutes or so, which is why infections can spread so quickly. Okay, I'll explain what viruses are a little bit later, but let's carry on with prokaryotic cells for now. So prokaryotic cells, those without a nuclear membrane, come in two varieties, the bacteria and the archaea. The former have been studied extensively, the latter much less so. Key difference is that archaea are much larger than prokaryotic cells. And some prokaryotic cells can perform aerobic respiration or use light as an energy source, so oxygen or light, whereas archaea cannot. Archaea, they basically live primarily, by eating bacteria, by eating bacterial cells. At least that's one of the methods they have for living. You'll find bacteria everywhere in pretty much every niche of this planet. Archaea tend to be poisoned by oxygen, so a lot of archaea you find in mud or in the silt at the bottom of oceans. I'm not going to go through this. This is basically the possible evolutionary pathway based on 16S RNA. RNA stands for ribosomal RNA, which I'm not going to go into now. But all cells, both prokaryotic and eukaryotic, contain small organelles, miniature structures known as ribosomes. And these ribosomes are where proteins are made. A cell cannot exist without ribosomes. Now, 16S is one component of a ribosome, one part of it. And it is made of RNA, known as ribosomal RNA, a type of nucleic acid. Now, by looking at the sequence of bases in ribosomal RNA, we can work out relationships between different types of prokaryotes. So we can see their relationships, their family tree. And what we're seeing down here is a sort of overview of the relationship as we think it stands now. But this is subject to change. This was a very long time ago that these creatures evolved. So a lot of this is guesswork, but very good guesswork based on molecular evidence. So classification of bacteria. Bacteria and archaea are so different that they are placed in separate domains. Life goes in domains. And bacteria and archaea are in separate domains, both prokaryotic, but different domains. Bacterial classification has been made systematic, but in practice is largely empirical. The primary distinction is based on the reaction to the Gram stain. There's a particular stain that you can put on bacteria, and it will either stain strongly or it will not. If it does stain strongly, the bacteria is known as Gram positive. If it doesn't, then the bacteria are known as Gram negative. Basically, the stain is staining the cell walls of the bacteria. So, Gram stain. Stain was invented by Christian Gram in 1884. differentiates on the basis of the cell wall. So as we can see here, we have got the gram-positive cell wall, and we have got the gram-negative cell wall. And the difference really is the peptidoglycan. Peptidoglycan is kind of like the bacterial version of cellulose. Plant cell walls, which are eukaryotic, use cellulose, a particular type of sugar, to give rigidity. Bacteria don't use cellulose, they use peptidoglycan. To break down that word, the peptid stands for peptide, meaning protein, and the glycan means sugar. So there is protein and sugar in the cell wall of bacteria. But the gram-positive have much more of it, so they stain much more strongly with the gram stain than the gram-negative do. And this has an effect because a cell that has more peptidoglycan in its cell wall needs to be treated differently with different types of antibiotic than generally than a gram-negative one. So there's a single layer of peptidoglycan about 50 to 200 nanometers thick. This is gram-positive. Other major component is something called teichoic acid. Some contain proteins or carbohydrates too. So this is what peptidoglycan looks like for any molecular biologist. But I'm not going to dwell on this because this is an overview of bacteria and viruses. So I'm not going to go too deeply into the detail. But this is the structure of peptidoglycan. Okay, so gram-negative, a double layer around 25 nanometers, commonly called an envelope, not wall. Inner layer is thin layer of peptidoglycan. Outer membrane contains phospholipid. Phospholipid is just lipid is fats, and phosphor for phosphate. So you've got phosphates with fats attached to them. Basically, phosphates are what we call hydrophilic. They like water. And lipids are hydrophobic. They don't like water. Fat floats on water. And you have a phospholipid bilayer in most cell membranes, well, in all cell membranes. And the phosphate part will point inwards towards the water of the cell or outwards towards the water around the cell. And the fats will point towards each other in a layer in the cell membrane so they can get away from the water. Lipoprotein, fat with protein, and lipopolysaccharide, meaning fats with sugars, multiple sugars, polysaccharide, many sugars. So there's an overview of the gram-negative envelope. So you can see the envelope here. There are the phospholipids, which are basically here. This is a phospholipid bilayer with the fatty tails touching each other to get away from the water and the phosphates pointing outwards. You have various proteins as well in the cell membrane, in the cell wall itself, which have all sorts of different purposes, which I won't go into right now. They give structure, basically, and some allow channels so that certain products can be pumped into the cell or out of the cell. So some bacterial shapes, we have rods, we have the cocci, clusters, we have spirals, and we have irregulars. We have a number of different shapes. Basically, we denote a lot of bacteria based on their shape. This is a generalized bacterial cell. So we have the pilus. I'm not going to go into the structure of bacteria too much right now, by the way. I'm going to give a second Zoom session on cell structure, but a quick overview of bacteria. They have these pili on the outside, which basically allow one bacteria to kind of link with another, and nucleic material can be passed between one bacteria and another. This is one way that antibiotic resistance can be passed from one bacteria to another. We have, in some cases, a capsule around the bacteria. The capsule protects the bacteria from temperature, etc. We have a cell wall where the peptidoglycan is. We have a plasma membrane where you'll have phospholipids. The nucleoid is the DNA, the big circular piece of DNA that contains the instructions to make new bacteria and for bacteria to survive. Cytoplasm is the liquid that everything floats in. Those little dots here are basically the ribosomes that make the proteins. And we have the flagellum. The flagellum is basically the tail. Not all bacteria have a tail, but many do. Many do. and this allows bacteria to swim. Some have one tail, some have multiple. Gram-positive and gram-negative. So here we have Staphylococcus aureus, cocci, a little round bacteria. Staphylococcus aureus is quite common. We have it in our mouth, we have it on our skin. It's normally harmless. It just kind of lives off our waste, so to speak, but it can become invasive if we have a weakened, compromised immune system, or if the bacteria finds its way into structures such as our heart. Okay, so gram-positive coccus in clusters. That's basically what a staphylococcus is. Neisseria gonorrhoea, a gram-negative coccus in pairs. So here we have the Staphylococcus aureus, gram-positive in clusters. And here we have Neisseria gonorrhoea, gram-negative coccus in pairs, just two little round bacteria together rather than in a cluster. The flagella, wall and membrane insertions. So this is basically the cell wall of the bacteria, and this is the flagella hanging out. There's a bunch of proteins here which allow this flagella to basically move in a kind of corkscrew action to swim. Flagella are basically protein structures composed of types of proteins known as flagellins. They're used for motility, rigid spiral shapes. You can have different types of flagella, as you can see on this image. Some bacteria have many flagella all the way around, some have many on one side, some have a singular one, and some have sort of polar at both ends. This is an actual image of these different types of flagella on bacteria. Fimbriae or pili, protein structures composed of fimbrins or pilins, 10 to 25 nanometers in diameter. Common versions for attachment to environmental structures. So basically, pili can be used to attach a bacteria to other structures, to tissue, for example, or two bacteria can join together to basically form a channel, and the pili can be used in a sort of sexual recombination, so to speak. Capsules, loose extramural polysaccharides or polypeptides, food reserves or protection mechanisms. So around the bacteria, you can basically get these capsules in some cases. And these capsules basically can protect the bacteria. The cytoplasmic components, the liquid within the cell, I'm not going to go into too much detail, but we have the mesosome. This is where the membrane can invaginate, so bend inwards. This allows bacteria to take in products. This invagination is quite useful. Ribosomes, they are the sites of protein manufacture, as I said earlier. The genome is a covalent closed circular DNA molecule, so a big circular piece of DNA containing all the genes of the bacteria, and with various inclusions within the bacteria, mostly for food stores. Endospores. Some gram-positives can make internal spores. These can survive environmental extremes such as high heat, desiccation, drying out, radiation, or extreme pH, acidic or basic. So these endospores can allow a bacteria to survive for prolonged periods of time in extreme conditions, and then emerge when the conditions improve. So distinctions between bacteria and archaea. A distinguishing characteristic between these two is the lack of peptidoglycan in the archaea. I think I'll say at this point that archaea is thought to be the parent of eukaryotic cells, so the ancestor of eukaryotic cells. Our cells in our body are eukaryotic, so plants or fungi, and archaea is thought to be the ancestor of the eukaryotic cells. We believe that an archaea, a couple of times in the past, fused with bacteria, absorbed the bacteria. The bacteria, some of the bacteria was aerobic bacteria that could use oxygen as an energy source, and when it was absorbed, it became what is now the mitochondria. Some bacteria were at a later point absorbed by Archaea, but those bacteria, they could basically use light as an energy source, and they became the chloroplast of plant eukaryotes. So eukaryotic cells are thought to be a fusion of Archaea and bacteria. So the walls are pseudomurin or S-form protein. That's what the walls are made of instead of peptidoglycan. Biochemistry and metabolism are very, very different from bacteria. So we're not going to spend too much time going into this. It's just showing pseudomurin, the structure, just to compare with peptidoglycan, which I showed you a little earlier. Here are some different archaeal forms. They come in all sorts of fun little shapes. And here's a few more. Distinctions between bacteria and archaea: metabolism. Crenarchaeota, hyperthermophiles. They can survive at 75 degrees C to 113 degrees C, utilizing sulfur. So these are a type of extremophile. They can survive in extreme conditions. Euryarchaeota, halophiles, 1.5 to 5.5 moles of salt they can survive in, basically. Methanogens found in environmental range from swamps to the guts of ruminants. Thermophiles optimum around 80 degrees C, some of which have no walls. So how do viruses fit into the 5 kingdoms or the 3 domains? Well, many people don't even consider viruses life, to be honest, because they cannot exist on their own. They have to live inside another organism, inside a bacteria, in which case they're called bacteriophages, or a plant or an animal cell, in which case they're just known as viruses. So in the 5 kingdom system, Prokaryotica covers all of the cellular organisms which lack a nuclear membrane. In the 3 domain system of Archaea, Bacteria and Eukaryota, the former 2 cover all of the cellular organisms which lack a nuclear membrane. Neither include the viruses because they assume cellularity. Basically, life is considered life if there are cells, and viruses are not cells. They don't have a cytoplasm. They're just a piece of DNA or RNA surrounded by a protein shell. Very simple structures that only really exhibit the qualities of life when they come in contact with the cell. They use a bacteria or a prokaryote or eukaryotic cells machinery to make copies of themselves. They're thought to have arisen within the bacteria or within the prokaryotes or within the eukaryotes that they subsequently infect as a sort of byproduct of the cell's normal metabolism that got out of control. But viruses are potentially in all 3 if lack of a nuclear membrane is the criterion. So it's a matter of definition. But biologists study viruses because it's more biology than chemistry, really, sort of. Are viruses living? If you consider that being a cell is the defining characteristic of life, then viruses are not living. For the purpose of this lecture, I'm assuming that viruses are living organisms. This is based on a scientific argument that reproduction is the defining characteristic of life. Alternatively, it is based on a non-scientific, sort of pragmatic argument that viruses have to be covered in biology because, well, you wouldn't study them in physics or chemistry or psychology, so let's stick them in biology. A brief bit of history of viruses. There are records of viruses throughout history. They've been with us as long as we've been with us. Here's a tomb painting in Egypt, about 3700 BCE. Shows a priest with typical signs of paralytic poliomyelitis. In 1196 BCE, Ramses V of Egypt died from a disease which seems to have been smallpox. Shows all the symptoms of smallpox. The symptom descriptions indicate that smallpox was endemic in China by about 1000 BC, so it was everywhere by that point. Well, not everywhere in the world, actually. It had not made it to the New World, to South America, or to North America for that matter. The Native Americans have got the Europeans to thank or blame for bringing smallpox to them, which they did not have a resistance to. Part of the reason that the Europeans found it relatively easy to conquer North and South America was because they took a number of infectious diseases, including smallpox, with them, which decimated whole cities and populations before a single shot was fired. On the 14th of May, 1796, Edward Jenner used cowpox to vaccinate an 8-year-old boy against smallpox. So basically, at the time, smallpox was endemic. It was there were regular epidemics of smallpox around the world. And Edward Jenner heard a milkmaid brag that she was never going to get smallpox because she'd already had cowpox, a disease that gives little boils on the hands. It was a common disease that milkmaids got. Edward Jenner was intrigued, so he found an 8-year-old boy that no one gave a damn about. And then he deliberately infected a cut on the boy's hand with cowpox from some pus from a milkmaid who was suffering from it. He then let the boy get over the cowpox and subsequently infected him with smallpox, which was lethal at the time in many cases. But the boy didn't get smallpox. And long story short, Edward Jenner had invented the first vaccine. And was sort of heralded for it at the time, although these days there would be a different response. Viral history too. So I'm going to mispronounce a few names here. I do apologize, but Ivanovsky, 1892, showed ceramic filtered extracts of diseased tobacco plants could transmit tobacco mosaic disease to other plants. This showed, because these were very filtered extracts, this showed that tobacco mosaic disease was not caused by some bacteria because it couldn't have filtered through these small pores. So there must have been some other thing other than just bacteria that could cause infections. Bejanek, 1898, extended Ivanovsky's results and coined the phrase contagium vivum fluidum, but also called the disease agent a virus, an ancient word literally translating as the word poison. Loeffler and Frosch in 1898 showed that a similar virus caused foot and mouth disease in cattle, a debilitating condition of cattle. Remilinger, 1902, showed that rabies was transmitted also by a filtrable virus. So rabies, another virus. Before that point, they thought it may be caused by bacteria, but nope, it was a viral disease. Landsteiner and Popper in 1909 showed that poliomyelitis was caused by a virus too. And Twort, 1915, and Dahel, 1917, discovered bacterial viruses, which they called bacteriophages. So if you ever hear the term phage, a bacteriophage is just a virus that infects bacteria. Bacteria can get viruses, prokaryotes can get viruses, so can eukaryotes. Plants, animals, everything can get viruses. What are they? Technically, they are known as obligate intracellular parasites. They're non-cellular, no metabolic activity, no mitochondria, no ribosomes, no way of replicating nucleic acids themselves, and no system for making membranes. They use the machinery of the host that they infect to make copies of themselves. They have no machinery themselves, just the instructions to make new viruses. The basic structure, there is a protein capsid composed of polypeptide subunits called capsomeres. So protein capsid just means a sort of shell around the virus. And then within that shell, you have nucleic acid. You can either have double-stranded DNA, dsDNA, single-stranded DNA, single-stranded RNA, or double-stranded RNA. DNA viruses tend to be quite stable over time. DNA doesn't mutate very, very quickly, so a DNA virus tends to remain the same for long periods of time. This is why if you had a measles vaccine as a child, measles is a DNA virus, it's probably still effective now as an adult, because it's composed of DNA and therefore doesn't mutate very easily. RNA viruses, however, such as influenza or COVID-19, although mutation rate of COVID-19 still remains to be seen, they are RNA viruses, and RNA mutates very quickly, which is why immunity to RNA viruses can in some cases only be temporary. Viruses can be enveloped, i.e. with an external membrane, or they can be naked, no membrane. The membrane can help protect the virus in certain conditions. COVID-19, influenza, sorry, not COVID-19, for example, is enveloped. Here's some human viruses. I'm not going to go through them, but as you can see, herpes virus, for example, is double-stranded DNA. We have got picornavirus, which is single-stranded RNA and naked. Some are enveloped, some are not. A naked virus is spherical, isometric symmetry. 16 nanometers, e.g. parvovirus, to 300 nanometers, e.g. pox viruses. Difference in sizes. They can be filamentous or have helical symmetry. I'll show you some pictures of these shortly. 80 nanometers to by 14,000 nanometers, e.g. the filoviruses. 15 nanometers by 400 nanometers, e.g. the tobacco mosaic virus. Here are some isometric arrangements of viruses, quite pretty. Naked viral structures, here's some helical, spherical complexes. You've got all sorts of different structures here. So you can see all the different types of shapes that viruses can actually come in. Here you can see, for example, we have DNA in the center. This is basically the sort of structure that HIV virus has. A base plate where it can bind to a cell and invade it. Enveloped virus is rare except for animal viruses. Most appear roughly spherical. Some have isometric capsids, e.g. HIV, lentivirus. Others have filamentous capsids, e.g. influenza. So there's some different viruses here. So we've got some COVID-19, we've got smallpox, we've got influenza. These are enveloped viruses. Basic steps of the life cycle of a virus. So first of all, virus attaches to a host cell that it's going to invade using specific receptor interactions. So for example, the HIV virus binds to the CD4 receptor, which all of our T lymphocytes, our helper T cells have, and therefore invades our helper T cells, which eventually disables those cells, and helper T cells help our immune system to function properly. Therefore, when HIV infects them and becomes symptomatic, turns into what we call AIDS, acquired immunodeficiency syndrome, the T lymphocytes, helper T cells, die, and the immune system therefore switches off. It's unable to fight infection. Therefore, an AIDS patient will die of all sorts of opportunistic infection. Okay, so they attach to a host using specific receptor interactions. They separate the nucleic acid, the DNA or RNA, from the capsid, the protein. Then within the cell, they make mRNA, messenger RNA, from viral information. It carries messages to the ribosomes, which can make proteins. Viral proteins are then made by the ribosomes within the infected cells. Basically new viral particles are made. The viral nucleic acid is replicated. Everything's packaged together, the protein shell and the new nucleic acid, viral nucleic acid, into new capsids. Now you have new viruses which can be released from the cell. They can either gradually be released from the cell, or they can cause the cell to pop, to rupture, releasing many viral particles to then invade other cells. So here's the basic envelope structure of HIV. I think I misled you earlier on, actually. I pointed to a different sort of structure for HIV. My mistake, wasn't thinking straight. But this is the basic enveloped virus structure for HIV. So we've got the nucleic acid in the middle, and we've got the protein shell around the outside. Okay, so this is the HIV life cycle, as we can see here. So this is a cell that the HIV is invading. So the virus will attach basically to a cell. It will penetrate the cell. Its protein shell will break apart, revealing its nucleic acid. Nucleic acid will go into the nucleus of the cell following replication. Certain genes will become active. So basically the nucleic acid of the virus will code for proteins to be manufactured. Here are the ribosomes that manufacture the proteins that will become the viral shell. Everything is assembled together, the viral proteins in the capsid, the nucleic acid within it, and then everything is released by a process called exocytosis. The viruses can then invade other cells. It's a life cycle of HIV and many viruses. Now, viroids might be the ancestors of viruses. We're a sort of simplified version of a virus. They're common plant pathogens. They're unusual infectious agents. They have a very small genome, single-stranded covalent circles of RNA nucleotides. No proteins are made. Potato spindle tuberviroid, for example, is an example of a viroid. So we basically just have a little circular piece of RNA that makes copies of itself. It's that simple. And that may be where viruses came from. They started off as these simple viroids, and they evolved through natural selection to eventually become viruses. Prions. Prions is where I'm going to sort of finish off here. Prions are basically proteins, no nucleic acid. They are proteins that can, well, sort of replicate. Scrapion is an example of a prion disease. Scrapie, a disease of sheep and goats, has been known about for several hundred years. Kuru is a disease of the New Guinea Fore tribe, first identified in the late 1950s. The Fore tribe basically were cannibalistic. They would eat their dead for a start, and if they ate the dead and the dead had a prion disease, then that prion disease would be passed to the living, and then the cycle would repeat. The Fore tribe, this disease was rampant in the 50s, but not in tribes that were not cannibalistic, which hinted to the cause of this disease. They are no longer cannibalistic. That is a banned practice in New Guinea. Creutzfeldt-Jakob disease, CJD, is another prion disease that can infect humans, can be passed over from cows. Bovine Spongiform Encephalopathy. These words can be difficult to pronounce, even for a biologist. BSC, first identified in 1986, and new variant CJD was described in 1996. So BSC 1986 and new variant CJD 1996. This is what prions look like. Mode of action is based on a change in conformation of a normal prion protein. This causes a buildup of insoluble protein fibers, amyloid in the brain, for example, of Alzheimer's victims. So this is a normal prion protein, and this is a diseased prion protein. Okay, thank you very much for watching this. By the way, here is my Twitter handle. If you were unaware of it, if you want to follow me, you can follow me at Mark of the D. That's a capital D, Mark of the D. Thank you very much for listening. And let's just see, just come out of this. Thank you very much for listening, and I look forward to speaking to you again soon.